Okay, I don't have too much in the way of announcements today. I still am planning on going over the questions that were most missed on your last exam. I'll have to do that on Thursday. Got one more person to take the exam. Kind of looking ahead, your next exam is not next week, but the week after. So I know we just had one, but when I test you more often, then I don't have to test you over as large amount of material. So in the long run, I think that works out best, even though I know you get tired of taking exams all the time. So it's only over 3 chapters. So, but just kind of keep that in your mind, try to keep up with things, keep up with your homework. Okay, so where we left off last time, we had finished up some of the physical ways to kill microorganisms like radiation. We explored the use of temperature, lots of other things, dehydration. And we started in with our chemical antimicrobials. So remember, I have these organized from kind of a low, medium, and high efficacy. I guess that's a little bit different than how the book has it organized, but the low level ones kind of work on more so the organisms that are easier to kill. Medium and the high level ones are considered sporicidal. So we talked about soaps and detergents. We'll move on to heavy metals, keeping particular in mind how we use these chemicals, if there's any big drawbacks or advantages to using them, as well as how they're going to kill cells that we're trying to kill. So heavy metals, basically they bind functional groups of proteins like that SH, that's a thiol group. Apparently proteins have lots of thiol groups on them, so that's where heavy metals bind and denature proteins. So I had kind of a hard time finding different uses for heavy metals. The reality is that we don't use them a whole lot because they're pretty toxic to us as well. So we certainly can't take them internally, not that we would any of these other chemicals as well, but they tend to be a little bit more irritating even when they're in contact with our skin. Probably the biggest use, at least traditionally, of use of a heavy metal has been in silver nitrate drops. That for years and years and years, they add silver nitrate drops to the eyes of babies almost as soon as they're born. Like even before the mother holds them, they put those drops in there. And the reason is because women that have gonorrhea, their babies during the birthing process are very susceptible to picking up that infection from their mothers. It often manifests as an eye infection that can lead to blindness. Okay, so to keep babies from contracting gonorrhea. I think there's 2 R's there, but I'm not sure. Okay, so they just do that for everyone, any baby born, because oftentimes women don't know if they have gonorrhea or not, or sometimes they wouldn't admit if they do. So gonorrhea can be asymptomatic in women, so they just kind of do that general for all babies. Now they've, for the most part, switched, at least in this country, to using antibiotic drops because they're a little less irritating to babies' eyes. Okay, another use of heavy metal, and by the way, what kinds of heavy metals? Mostly we're talking about copper and silver. Mercury is antimicrobial as well, but obviously mercury is even more toxic than copper or silver. So we're mostly thinking about those 2 heavy metals. Copper sulfates has some use as an herbicide. If you've ever been driving out in the country and you see a pond that's totally covered with green scum, okay, they could get rid of that by adding some copper sulfate to the pond to kind of cut down on the algae growth there. Some fish are sensitive to that as well, so it depends on what kind of fish are in the pond. If you use too much, it can kill the fish. So have to think about that. Actually, if you throw a penny in a fish tank, that can be enough copper to actually kill your fish. So again, fish are somewhat sensitive to copper. A few years ago, they came out with a copper-based hand gel that was supposed to be less irritating than the alcohol-based ones that we're all so familiar with now. I couldn't find any of that on the market anywhere, so I don't know if those are still available or not. So again, not a lot of uses of heavy metals. You do use them in very small amounts, and they do target all cell types, of course, not endospores. One other use I've seen on the market, cleaning rags that have silver infused in them. I don't know how effective those are, but again, that's because of the antimicrobial properties of silver. I think you can get socks with silver in it. It's supposed to cut down on odor, because the odors come from bacteria on our feet. Okay, like I mentioned, not as many uses as we'd like because of the high toxicity. At one time, there were bandages on the market that had silver in them. Again, just a little bit more irritating. I haven't been able to find them anywhere anymore. Allergies are pretty common. If you may have noticed you have a metal allergy, if you have certain kind of earrings that you can't wear, that your skin gets irritated, for instance. And there is some resistance out there even to heavy metals. So not as many uses. I did kind of an interesting little experiment a couple years ago. I had a lawn of bacteria that I think it was bacillus that I put in the lab. And interesting enough, before 1982, pennies were made of 95% copper. But after 1982, because it's cheaper, they switched over to pennies being almost all zinc, 97% zinc, and then just coated it in copper. So I did a little experiment. I cleaned a couple of pennies with bleach and then dried them off and put them on that lawn of bacillus and incubated it. And you see this mostly copper penny had a really nice zone of inhibition around it. I don't know where exactly you would measure that, but definitely had some bacteria being killed by that copper. The newer penny that was mostly zinc, we definitely had a zone here, but also we've got some colonies growing right under the penny. So it would at least appear from my one sample size experiment that copper is a lot better antimicrobial than zinc. Okay. All right, so phenols are kind of considered a step up, maybe somewhere between low level and intermediate level disinfectant or antiseptic. Phenol is another one where I really had to kind of struggle a little bit to find some uses for. It's fallen out of favor using phenol just because of some toxicity, irritation issues. But pretty much phenols have in the past been considered kind of the gold standard antimicrobial. Like that was the go-to to use if you needed to cut down on any antimicrobial growth. Because it does it all, disrupts the cell walls, membranes, denatures proteins, just a lot of damage to the cell. A little feedback or something maybe from the speaker up there. Okay, so why was it considered the gold standard? Kills all sorts of organisms, not just bacteria, but viruses, fungi, all sorts of things. And it does pretty well when the organic load is high. So if you're trying to disinfect a dirty surface or something that's been contaminated with blood, for example, phenols actually cut through that pretty well. And they work for a while. Okay, they maintain their activity over time, and they're pretty cheap. We can use them in low concentration. Another nice thing is that mycobacteria are susceptible. Phenol penetrates really well through that acid fast, that waxy cell wall. So that's another advantage. So this is just straight phenol. What this is a just what a phenol would look like. That phenol by itself, like I said, is fairly toxic. We don't use that at all anymore, but we have found that if we kind of monkey with the structure of phenol a little bit, add some side chains, for example, then that can maybe cut down on the toxicity a little bit. So we call these phenolics when we add some side chains to that. This orthocreosol is basically kind of an intermediate in a lot of industrial processes. A bisphenolic is with 2 phenol rings and a chlorine attached here. This chemical triclosan used to be in all sorts of things. We put it in plastics, high chair trays, plastic trash bags, cutting boards, all sorts of things, until it was outlawed a few years ago because we found that we were able to detect triclosan in people's urine. Okay, so it was touted as being safe for a long time, but then we started to have concerns. Well, how safe is it if it's building up in our bodies? So mostly we don't see that anymore. There's big controversy over that for a long time. Orthophenol phenol used to be the active ingredient in Lysol back in the 80s, but we since again switched to an active ingredient. I think it's a quaternary ammonia that's actually the active ingredient in Lysol now, and just a little bit safer. You may remember back when we talked about this guy, Joseph Lister. That was probably back like the first day of class. If you remember, he was the one that advocated spraying carbolic acid. Carbolic acid is phenol in the operating rooms as a mist. Doctors wash their hands in phenol. They, rubbed it on the person's body pre-surgery, and that cut down a lot on surgical infections. Unfortunately, it had some side effects. Doctors' hair fell out, their skin bleached white, their nails cracked. They had respiratory issues from breathing this in. So we don't do that anymore. Again, this triclosan used to be found in all sorts of things, even toothpaste. All right, once an industry starts lobbying that, hey, this is an antibacterial, this should be required to be in certain things. That's the way it was for a while, not anymore. They even had some fancy trade names for triclosan, like BioFresh. How could something be called BioFresh and not be good for you, right? Okay. And we're also finding, though, that some organisms like Pseudomonas were developing some resistance to triclosan. So, okay, again, heavy metals, phenols, not in as much use today, certainly. Halogens, if we move up to the intermediate level, you can think of lots of different ways that we use halogens. So if you think about the periodic table, the halogens are off to the right-hand side, chlorine, iodine, bromine, and fluorine. We are mostly going to focus on chlorine and iodine. Remember, the halogens have an outer shell that's almost full, so they like to grab electrons to fill up that outer shell. So they're very reactive, and they're considered strong oxidizers, right? They're grabbing electrons from other molecules to fill up their outer shell. So causing especially a lot of cell damage, but especially denaturing of proteins. We are, like I said, we're going to focus mostly on chlorine and iodine, some uses of bromine. I hear now of bromine being used in hot tubs, especially because it doesn't evaporate as quickly as chlorine. Maybe a little bit less irritating than chlorine. Of course, we put fluorine in our water supply. It helps us keep us from developing tooth decay, disrupts biofilms in our mouth. So that's, most people would argue, a good thing. And advantages of the halogens, they work fairly quickly, within 30 minutes, usually a lot quicker than that. So I'm not sure where I got that number from originally. They're not considered sporicidal because it takes several hours to kill spores. So to be considered sporicidal, it has to do it, I think, within like 10 minutes to be able to deactivate them. Okay, so let's talk about chlorine first. Okay, you're all familiar with using chlorine probably at some point in your life. It's a pretty strong disinfectant. Remember, something that's a disinfectant targets non-living surfaces, or it's designed to be used on non-living surfaces. You could get a little bleach on your hands and it's not going to hurt you. You wouldn't want to do that, you know, over time every day. That would be pretty irritating to your skin. It is susceptible to organic load, so that means you have to clean something first and then bleach it to disinfect, to reduce microbial numbers. And it's pretty sensitive to light and pH. So when you buy bleach, you're always going to buy it in one of these opaque containers, because the more it's exposed to sunlight, the quicker it goes off. Basically, it dissociates into water, and it's not going to be effective anymore. And even, I always try to buy bleach in pretty small bottles, because after about 6 to 9 months, your bleach is not going to work as well. Even though, again, it's protected from the light somewhat, it starts to dissociate and not work as well. So it won't have the same killing power if you've got a bottle of bleach that's been sitting around for a couple of years in your cupboard as one that's fresh. So keep that in mind. Won't make your whites as white either. So keep your bleach as fresh as you can. And of course, in a swimming pool, you got to keep the pH from getting too high. It won't, again, work if the pH is too high there. So I'm not too concerned that you are familiar with the exact uses of the different forms of chlorine, but we do see it in different forms. The elemental chlorine is often what's added to the water supply. It's kind of a final stage of cleaning of, you know, wastewater treatment, where we dump it back out into the environment. That's why you see those big ponds at wastewater treatment plants, because they have to give the water time for that chlorine to evaporate off before they release it. Chloramine is actually also sometimes used in water treatment. It's a liquid form of chlorine and ammonia. So you may have heard, you know, somewhere, somebody telling you at some time, never mix your cleaning products. If you mixed bleach with ammonia, that gives a toxic vapor that can kill you. So it seems weird to me that we can put those two things together, bleach and ammonia, in a liquid form. It's okay as a liquid form. It's just when it makes this vapor that it's toxic. And sometimes we soak bandages in chloramine. There's some uses there. Hypochlorites, that's what bleach is. So typically in lab, we use a 3% hypochlorite or 3% bleach in order to wipe down the benches. This semester, we switched to using alcohol, just so we could spray it over a large amount of area. Okay, and then you can use chlorine as a gas as well if you need to disinfect some kind of large space. All right, and then our next halogen, iodine. Not making you write quite as much today, just more kind of delivering information. And I was kind of looking at my organization here. I realized that I didn't really tweak this slide as much as I would like to. I feel like this information is kind of disorganized, but bear with me here. What I want you to know about iodine, it's a little bit better when there are organic presents, so it's a little less susceptible to organic deactivation than what bleach is. We use it in several different ways. Sometimes we dissolve it in water, and we just call it iodine. Sometimes we dissolve it in alcohol, and then it's called a tincture of iodine. So you might have heard that word before and wondered what that meant. A tincture is anything that's dissolved in alcohol. It's supposed to be a little bit more effective little bit more penetrating when you dissolve it in alcohol. Uses of iodine, wound treatment is one that we traditionally use that for. Probably it's a little bit more often used in kind of pre-surgery, or pre-needle poke, you know, wiping it down on a person's skin before surgery. Again, letting it sit there a little bit, make sure give enough time to do its thing. You can use iodine tablets. So again, I have this under like tincture of iodine. I don't think you would use it. I think you use actually iodine tablets to put in water. It's a really lightweight thing to carry with you if you're backpacking. Just carry some tablets, you add it to some water, you let it sit for a certain amount of time, and then the water is supposed to be safe to drink. Unfortunately, the iodine is not good at killing some of the protozoa that might be in the water. So it's better than nothing, but it's probably better to filter the water if you want to drink it. Like cryptosporidium cysts may survive the iodine treatment. It does make the water taste a little funky as well. And just with the use of iodine, there is a little bit of irritation to the skin when I use it as an antiseptic. If you use it on surfaces or your skin, it's going to stain it for a little while. So that may not be what you want. There are, you know, some allergies, cause some rashes with some people. Probably a more popular way to use iodine is as an iodophore. Okay. And that just means that iodine is complexed to some kind of a protein, usually a protein carrier molecule. Okay. And that tends to give the iodine a little bit slower release. So it's going to be longer acting over time. It tends to be a little bit less irritating to the skin. Okay, so the trade name for that an iodophore is betadine, right? And that's probably a form of iodine is more likely to be used again before a needle poke or in some kind of wound dressing. Okay, here it looks like their example from the book is they're washing a patient's hand in some form of iodine, I think it's betadine, before a hand surgery. Okay, another application, dairy applications. So I don't think I posted this picture, but there's such a thing, and it's a little crude sounding, it's called teat dip, right? So it's one more way to try to make the milking process as hygienic as possible. This is like the cleanest cow I have ever seen. Wouldn't think you would need it. But basically, they dip it on the teats before they milk. They go down, they do this for like 4 or 5 cows in a row, give it a little time to sit there, then they go back down and they wipe it off before they actually do the milking. Okay. All right, so still in that intermediate range, alcohols. So I'm talking about the use of ethanol and isopropanol. Probably the big use that we're all familiar with now in terms of using alcohols is in an antiseptic hand sanitizer. Okay. It can be used as a disinfectant as well, usually a little more concentrated ethanol or isopropanol. Isopropanol is rubbing alcohol. I always use isopropanol to disinfect thermometers after I use them. But we do use different concentrations, and typically we might think, well, the more concentrated we use the alcohol, the better. That's not necessarily the case because some water is necessary for the alcohols to do their trick for the surfactant action. Okay, so one big thing that alcohols do is dissolve membranes. Okay, so that's surfactants, it's breaking apart membranes. Okay, also denatures proteins. So, you know, is 80% necessarily more effective than 70%? I don't really know. But it appears that some water is necessary. You wouldn't want to use 100% alcohol because there's no water in it, and it would evaporate really quickly, 100% alcohol. So the water keeps it from evaporating as quickly as well. I was looking, I had about 4 different brands of hand sanitizer at home, and they varied from about 60% ethanol to 70% ethanol. So depending on the brand, Purell usually has a little higher concentration of alcohol than ethyl alcohol than what the Meyer brand does. I've noticed the ones that you see all over campus, these are 80% ethanol. So still considered, again, an antiseptic, safe to be using on our skin. Okay, so it's pretty non-irritating to the skin. It's pretty cheap. Alcohol is considered an inhalation hazard, but what you inhale from using hand sanitizer is just a minuscule amount. So we don't have to worry about that. If you inhale too much of it, it can affect the nervous system. So again, have to just keep that in mind. So I thought it was a good time in the midst of our pandemic to think about good ways to wash our hands, or the best way to wash our hands. And if you've been trained in a hospital or whatever, I'm sure you probably know this already. But if you had your choice of whether to use hand sanitizer or wash them with soap and water, what's the best thing to do? Well, if you're going to use both of them correctly, okay, you're going to use enough to be wet for about 15 seconds. Oops. Okay, so you got to use enough hand sanitizer to keep your hands moist for about 15 seconds for it to do its work. And if you're going to wash your hands, they say you should lather up and scrub for about 20 seconds. It's probably a little longer than what I'm used to doing. So maybe I could do a better job at washing my hands. The other thing to remember is that we talked about, I'm not sure if I mentioned this on the previous slide. I didn't mention this. Alcohol is actually not very good at penetrating through organics. So that's something to take note of. Does not penetrate well. Presence of organics. Okay, and that's something definitely to keep in mind. About 5 or 6 years ago, I went on a trip to Guatemala, and we were kind of out in the back country building houses. And we'd stop for lunch and we'd pass around the hand sanitizer and think that, you know, oh, we're doing a really good thing before we eat. We're getting rid of, you know, all the bad bacteria that might make us sick. And reflecting now, probably that hand sanitizer wasn't doing a whole lot of good because our hands were dirty. We were, you know, building in the dirt basically, and that might actually not have been doing a lot of good. So the other thing about hand sanitizer, it does not reliably work on naked viruses. Okay, so most viruses that make us sick are enveloped, and it works really well on enveloped viruses. So like coronavirus, flu virus, hand sanitizer works really well. But something like norovirus, it doesn't work as well. So if you have an outbreak of that, then you're definitely going to want to wash your hands whenever possible instead of using hand sanitizer. So because of these two drawbacks of hand sanitizer, usually washing hands is best. Just that kind of washing away action is really good for getting rid of a lot of things that would make you sick. Hand sanitizer is good when you can't be washing your hands all the time. So don't give it up. You know, after you go to the store or whatever, I like to wash off with a little hand sanitizer just to be safe. Okay, let's move up to our high-level disinfectants and antiseptics. Let's start with hydrogen peroxide. Okay, if, we talked about, different things to think about when you're choosing an antiseptic or a disinfectant, and that there's no perfect disinfectant. I've read that hydrogen peroxide is really the closest thing we have to being a perfect disinfectant. At high concentration, it's considered sporicidal. So it's even killing spores that are pretty difficult to kill. Lower concentration, it's safe enough to be used on our skin as an antiseptic. Why does it kill when hydrogen peroxide, remember this is H2O2, when this decomposes, it decomposes into hydroxyl radicals. Okay, so this is an OH with an unpaired electron. This is the most toxic of all those toxic oxygen compounds that we have talked to. It's grabbing electrons away from everything. It's a very strong oxidizing agent. So it's doing everything. It's denaturing proteins, breaking down membranes. Okay. One disadvantage is that, like bleach, it's photosensitive. So whenever you buy hydrogen peroxide, so it's going to be in a dark bottle like this. I found this in the back of my medicine cabinet. The date on it is 1995. It was totally evaporated. There wasn't anything in it, and I found it in my medicine cabinet. But that opacity is supposed to keep the hydrogen peroxide from going off. We used hydrogen peroxide in lab last week. If you're in lab for our catalase test, catalase is an enzyme that detoxifies hydrogen peroxide into water and oxygen. So you're looking for those oxygen bubbles. We have to make up hydrogen peroxide new every semester, because if it starts to go off a little bit, decompose a little bit, it won't work. The catalase test doesn't turn out the way it's supposed to. But advantage is it doesn't leave any residue on the surface that you're trying to clean. It doesn't damage surfaces at all. It's very safe for us. Okay, so we usually use 3% as an antiseptic or a low-level disinfectant. So back when I was a kid, if I would skin my knee, Mom would bring out hydrogen peroxide and you spray it on. It didn't really sting very bad, and you'd see lots of bubbling as a result. That both our cells and bacteria cells are making catalase and bubbled up. If you're going to use it as an antiseptic, you have to use enough so that it stops bubbling. So you have to overwhelm the bacteria's ability to produce catalase to detoxify it. And then it works really well. We almost never use it anymore as an antiseptic because if you use it repeatedly on a wound, it will slow down the healing process because it just slows down our own cell regeneration. So it's more likely to leave a scar if you're using it repeatedly. First time it happens, that's great use of it, but then you should switch to using something else, like some antibiotic cream that's going to be, again, not hindering the healing process. We can use it as a little higher concentration as a liquid or even as a vapor, especially for sterilizing instruments that you can't put in the autoclave that would be sensitive to heat. This is a really good tool. Okay. Probably chemicals that you have a little less contact with are the aldehydes. It's another high level. They're always used as disinfectants. They cross-link proteins and DNA, so doing a lot of cellular damage. Another good thing about them is that you can use it in the presence of organics, and they're not going to be corrosive to delicate medical equipment. Huge drawback is their toxicity. They're even in some forms considered carcinogenic. So that's really going to limit their use. But most commonly they're used in the form of, I wrote it down here, glutaraldehyde. That's a little bit more expensive form of an aldehyde, but it's a lot safer to us. Again, sensitive medical materials or cutting tools can be soaked in glutaraldehyde and then sporicidal. So effective way to sterilize equipment. Another form, formaldehyde. That's the form that is a little bit more toxic to us, considered cancer causing. Typically, we use that in a form called formalin, which just means formaldehyde dissolved in water. That's a common preservative, or at least it has been in the past, for embalming. Our cadavers here at Grand Valley are preserved in an alcohol solution, so not formaldehyde, alcohol. Some vaccines use a tiny, tiny amount of formalin in order to deactivate the virus or deactivate the toxin, so that then that deactivated virus can be used to stimulate our immune system. So it's such a small amount that it's considered to be safe. They used to put mercury in some vaccines as a preservative as well, and they don't do that for any that are given to kids anymore, at least. Okay, one more chemical that I want to talk about, and that's just a few different gaseous agents like ethylene oxide or propylene oxide. Again, lots of cellular damage, going to cause some mutations, going to be acting on proteins as well. We would use a gas most often to sterilize some object. Maybe some object that was too big to be put in an autoclave, like a mattress or something like that, that an ill person was sleeping on. Of course, my picture here is of a really small contraption here. Basically, you could put some medical instruments in there, flood it with the gas for a certain amount of time, and then it would be sterile. The main drawback here, you got to know what you're doing when you're working with these gases, because they're explosive. So you usually have to add some carbon dioxide or nitrogen at the same time. You have to basically blow off, you have to flood the object with some inert gases to get rid of all the gas when you're done. But it's really good, especially for some sensitive medical instruments like a heart-lung machine or an endoscope. So there was an outbreak of some antibiotic-resistant bacteria on an endoscope about 10 years ago. Really small instrument with tiny little grooves in it. You can't autoclave something that's so sensitive like that. And it's hard for chemical disinfectants to kind of get in all the cracks. So they found that sterilizing with a gaseous agent like ethylene oxide was the best way to go for something like an endoscope. Okay, comments, questions on any of our chemicals? Okay. All right. So it's a little segue into talking about antibiotics. I think that I posted this chart as well, just because, not because I want you to memorize all this, but I think it's extremely interesting. This chart was published in the New England Journal of Medicine a while back. And it looks at the things that used to kill us a little bit over 100 years ago in 1900, and kind of more present day 2010, the sorts of things that we die from. So if you look at the things from 1900, the first few causes of death were all infectious disease. So number one, pneumonia or influenza. Of course, this is even before the big pandemic in 1900. So that's kind of surprising. TB, very successful pathogen, thousands of years. So that's interesting. GI infections. So that's going to be things like cholera or typhoid that we don't see anymore in any developed country because we treat our water. Then we get to a few things that we still deal with today: heart disease, cerebrovascular disease, basically that means strokes, kidney disease, accidents, cancer, senility, and here's another infectious disease, diphtheria. Okay, so if you compare those things to what we see today, you'll notice that there's only, let's see, a few infectious diseases on here. Pneumonia or influenza made the list there. I guess that's the only one. Mostly things that you die of when you're older. Non-infectious airway disease, that's something like COPD or emphysema. Stroke, well, accidents, of course, can happen at any age, but Alzheimer's, suicide made the list here. But you also notice that we're dying less often in 2010. Okay, so the life expectancy has increased a lot. In 1900, for a white male, life expectancy was, whoops, not 1947, was 47 years. Because in 1900, about a third of kids died before their 5th birthday. So when you see the life expectancy of 47 years, that doesn't mean people lived till they were 47 and they died. That's kind of a misconception. People still lived, to 80 or 90 years old, but so many kids died, their 1st or 2nd birthday that it averaged out to about 47 years. So that's pretty striking. Again, so many infectious disease there. In 2010, the life expectancy was more like 74 for a white male. Okay, so made a lot of progress there. Why have we made so much progress? I really like this slide, and I wish it was a little bit more updated. Especially, it would be interesting to have figures from this year. But if you look at the sort of things, again, this looks at the death rates, and it kind of superimposes kind of what was happening in the world that might have influenced the death rate. So anybody's interested in public health, this is the sort of stuff that should be interesting to you. It makes note of beginning of health departments in states here about the year 1900. Here's a big jump forward in hygiene when we first started to chlorinate, treat our water. Okay, so our hygiene is getting better and better. We had this huge jump, of course, during the worldwide flu pandemic, 1918, 1919. But other than that, you'll see the death rate steadily going down. We had the last human-to-human transmission of bubonic plague. This was before antibiotics came out. So when we understood that basically you clean up your trash, you don't have as many fleas, and you're not going to have the transmission of plague. So getting better hygiene influenced that. And then here, mid-40s, first use of penicillin. Now we have drugs to treat our infectious disease. And so hygiene, antibiotics, kind of the third piece of the pie here was vaccines. So the Salk vaccine, that was one of the 2 vaccines that were developed to treat polio. So got more and more vaccines here as time went on. We see a little uptick here after 1980. I'm not really sure. I don't think it kind of explained that. Could be due to antibiotic resistant infections. We're on the up. People are vaccinating less. I think people are doing a little bit better job of that now. That's been in the news a lot. Also probably AIDS influenced that as well, an infectious disease that we weren't good at treating until maybe in the last 10 years or so. So, all right, so just kind of thinking holistically a little bit before we talk a little bit more specifically about antibiotics. All right, so where are we going here with chapter 10? I always like to talk a little terminology first, but then we're going to talk a little bit more history. this is something that your book doesn't talk about at all, but I think that it bears mentioning what kind of organisms produce antibiotics for us. Just like we talked about what things we have to consider when picking what kind of disinfectant to use, what do doctors have to keep in mind when they're prescribing antibiotics? How should they be administered? What kind of things do they target? What they target is what we're going to spend a lot of time on, what we call the mechanism of action, how antibiotics work. What do they target in bacterial cells? And then we're going to run through about 15 different antibiotics and look at them in a little bit more detail. So the reading in your book for this chapter is not very long. It's only a few pages because your book's coverage of antibiotics, they talk about sort of like all the general information about antibiotics, and then they have pages and pages of tables listing a bunch of different antibiotics. Okay, so you don't need to memorize all those tables. It's not a pharmacology course. We're going to talk about 15 kind of and ones that I've picked, ones that hopefully you'd be a little bit more familiar with to focus on. Okay, and then we're going to mention antibiotic resistance. So really hot button topic, a little bit more about how that actually works. How do bacteria acquire the ability to survive in the presence of antibiotics? All right, so a little terminology that I'd like to start with, just so we're kind of clear on terminology. A chemotherapeutic drug. Usually when we think about chemotherapy, we're talking about cancer, but if you use that term correctly, any chemotherapeutic drug is any drug that's used in the treatment of any disease. So it could be insulin, it could be a cancer drug, it could be an antibiotic, all those things. And then an antimicrobial agent is kind of a umbrella term using that word to, any infectious agent. So it could be a drug to treat a protozoa infection or a viral infection. And then by strict definition, an antibiotic is an antimicrobial agent that's produced by organisms. So it's just naturally secreted by some kind of organism. We'll talk about what kind of organisms. And it targets bacteria only. Okay. So an antibiotic is not going to work against a virus. And then let me get my words straight here. A semi-synthetic drug is an antibiotic that's been modified a little bit. So I know that term is a little confusing. It kind of doesn't sound like an antibiotic, but usually that's the way it's used, a semi-synthetic. So we've taken the natural product of some organism and monkeyed with it a little bit in the lab to maybe give it a little bit more efficacy or make it last longer or make it less resistant, something like that. And then a fully synthetic drug, that's an antibiotic that's been synthesized in the lab. Most of the antibiotics that we use today are either true antibiotics or they're semi-synthetics. A sulfa drug would be an example of a synthetic drug. We don't use those a lot anymore, but we'll talk more about that. Okay, so if you use these terms in sort of the strict sense, we could split hairs there a little bit. All right, discovery of antibiotics. I added one little picture up here yesterday, just because I felt like I was leaving him out. We'll start out, we mentioned Paul Ehrlich before. He was kind of considered the father of chemotherapy. He's the first one. We're talking, turn of the century there, 1900s, early 1900s. Really kind of think carefully about treating infections and screening large number of chemicals to try to find a drug that would work on a particular infection. He actually was trained in Robert Koch's lab in Germany. And he found an arsenic containing chemical that worked against syphilis. Had some use. Unfortunately, arsenic is really toxic to our bodies as well, so it had limited use. But he used that term magic bullet. Magic bullet. Okay, I think I spelled that right. A magic bullet would be one that killed your enemy and not people on your side. So he was looking for drugs, again, that would target bacteria and not harm our cells as well. Then really no other chemicals were found to be effective against infectious disease until the 1930s, where Gerard Domach, another German scientist, and this is him right here. That's the picture I added. You can just tell that he's German just by looking at him. I don't think that little mustache is ever gonna come back in fashion. But again, this is right around the time of, right before World War II. He was screening a large number of sulfa-containing chemicals to try to find one that worked against streptococcus. Okay, and he found one. He called it Prontosil. So you may have heard that. He won the Nobel Prize in medicine in 1939. Hitler wouldn't let him go and accept the prize because he was mad at the Nobel Committee for who they named as their Peace Prize winner a few years earlier. So he did end up getting to accept the prize eventually when the war was over, but he didn't get the money for it, unfortunately. Sulfa drugs were a miracle in the 1930s when they came out. They had very limited effectiveness. It was mostly streptococcus that they worked on. They worked on strep throat, scarlet fever. So credited with saving thousands of lives, including Winston Churchill in the 1930s. So how important he became. In 1929, though, is actually when penicillin was discovered by Alexander Fleming. I wanted to say Hamilton, and I knew that wasn't right. So here's Alexander Fleming looking very scholarly in his lab, his hospital lab in London. So the story goes, he went away on vacation, and when he came back, he had left back in those days, Petri dishes were made of glass, and it was somebody's job to clean them out, but they never got done. He had a bunch of Petri dishes over in the corner of his lab that he realized didn't get thrown away. Came back from vacation, and he noticed this big mold, like right here, growing on his plate. He'd been doing research on Staphylococcus and ways to kill Staphylococcus with an enzyme called lysozyme that he discovered that breaks the bonds between those sugar units of peptidoglycan. We've talked about lysozyme. But he noticed there was a mold growing on his Staph aureus, and that mold was inhibiting the growth of the Staph aureus. Chance favors the prepared mind. He realized that mold was secreting something that was killing the bacteria. So he actually was able to identify the substance. He called it penicillin, that Penicillium notatum mold was secreting, but he wasn't able to figure out how to mass produce it. So he wrote it up in a paper, then he moved on to research something else. About 10 years later, also in England, working at Oxford, Howard Florey was looking for ways to, he knew the war was coming, and he was looking for ways to fight microbial infections because so many people die on the battlefield from infection rather than bullets. And he was looking through the literature, and he found this paper by Alexander Fleming and he assembled a whole lab of people to try to figure out how to mass produce penicillin. So here's Howard Florey. He worked together. Ernst Ch9 was a organic chemist that he got to help him figure out, again, how to mass produce penicillin. And it was a very difficult task. They ended up moving to a lab in the US. They found a little different strain of penicillin that they isolated from a moldy cantaloupe. They found they could get the most drug isolated from that. And it became available, within, to everyone near the end of World War II. They fought about whether to patent this drug or not, and they ended up not. So it's pretty astonishing. That would never happen today. It's interesting, the whole vaccine issue. Companies are poised to make a lot of money out of that, even though the government has given them money to do that. So times change. Okay, so where do antibiotics come from? Your book doesn't address this at all. We talk about it a little bit in the lab manual on that lab that we talked about antibiotics in lab 12. So I thought I would mention it here again because some people have misconceptions about this. But all the antibiotics that are in use either they're secreted by a bacteria or a fungi. Okay, so we don't get antibiotics from, platypuses or something like that. They're the natural product of one of those two kinds of organisms. Why do fungi secrete bacteria or antibiotics? We don't really know. Initially, it was assumed, well, it helps that organism outcompete in its environment. But we think now that they don't produce enough antibiotic in the environment to actually kill other organisms. We can make them produce more in the lab when they're on this nice growth media, but in the environment, they just don't make very much. It's a question of, well, why did they make it then? They must spend a lot of energy to do it. It might have some kind of signaling mechanism. We're not really sure. Okay, but just some examples of some genera that make a lot of medically useful antibiotics. Bacillus, we use some bacillus in the lab. Bacillus are very common soil organisms, along with streptomyces. Again, these are soil bacteria. About half of the antibiotics in use now come from streptomyces. So it's incredibly useful bacteria for us. It's a unique bacteria because it actually grows in long filaments and produces reproductive spores, kind of like fungi. But these are prokaryotic cells. They don't have a nucleus. So they're still considered bacteria. And then the fungi, of course, you've heard of penicillium. Sometimes those are a little bit greenish molds there. And another common one that's given us a lot of medically useful antibiotics is a genus called Acremonium. A few years ago, that we changed the name of that genus. It used to be known as Cephalosporium, which made a lot more sense because we have a whole bunch of drugs that are called cephalosporin drugs. But I don't know, for some reason, they changed that genus name. So that's just some examples. There are many more. Okay. So some things, again, that we need to think about when using antibiotics. All right. The number one thing that we have to keep in mind before any antibiotic is used, is it selectively toxic? Okay, so that means, does it target a pathway or structure? Of microbes that hosts lack. Okay, that's the most important consideration in using an antibiotic. Does it target bacteria, and is it safe for us? You might have heard it said that we're running out of antibiotics to use because of the resistance problem. We're actually developing a lot of antibiotics. The only problem is almost all of them are too toxic for us to be able to ingest. Okay, so that's a huge issue. It's the absolute first thing. If it's not safe for humans to take, then it's not going to be worth anything to us. Okay, have very little value. Okay, so I thought I'd put that on a slide all by itself. So what sorts of ways are drugs selectively toxic? Well, this is the graphic from your book. Actually, this is from the 5th edition book. The 6th edition made a few changes in this. When they changed the 6th edition, they didn't give instructors access to all the new graphics in this chapter. So I've got the old one up here still, but it's pretty similar. So if we look at mechanisms of actions, this is really what we're going to spend the most time on. If you know anything about the specific antibiotics we talk about, you're going to want to know how they work, their mechanism of action. So the most common mechanism of action by far is this inhibition of cell wall synthesis. Okay, because think about it, our animal cells do not have cell walls. All right, so if it's targeting the cell wall, it should be very safe for us to take. Sometimes students will make a mistake and say, oh, eukaryotes don't have cell walls, which is not true. Remember, plants and fungi have cell walls. It's animal cells that don't have cell walls. Okay, so we're not going to worry about anything that's an antifungal. We're going to focus on drugs that work on bacteria. So here's some examples of ones that we're going to be talking about a little bit later. The next biggest category is drugs that inhibit protein synthesis. Because remember, bacteria have a slightly different structure on their ribosomes. They're 70S ribosomes instead of our 80S ribosomes. We get a little more side effects from those drugs typically because the structure is similar, but different enough that we can exploit that. Let's see, we're not going to worry about the antisense nucleic acid ones or the chloramphenicol, but we will be mentioning those other ones. Disruption of the cytoplasmic membrane. Not as many drugs, we don't have as many drugs that can do that because bacterial membranes are very similar to ours. So we have a lot of toxicity issues there. The cell membrane inhibitors that we do use, we can only use on our skin. We can't ingest them because they're just too toxic to us. An example of a drug that inhibits some metabolic pathway that we don't have, that's how the sulfa drugs work. So we're going to look at that in more detail a little bit later. Not familiar with dapsone. It might be an antifungal, I'm not sure. And then this is where in your book, they have these as two separate categories, drugs that inhibit RNA synthesis and drugs that inhibit DNA synthesis. Here, they're just together in one category. We're not going to worry about the first two here. We're going to talk about the quinolones and rifampin as examples there. They're targeting enzymes that bacteria have. They're a little bit different than the ones that we use. And we're not going to worry about this last mechanism. These are antiviral drugs that might inhibit a virus from attaching to a host cell. In general, we don't have very good antiviral drugs. Probably the exception being for HIV. We have some good drugs that work for HIV, but you know, flu, we got drugs for that, but they don't work very well. Okay, and of course, we're searching for things that would help with coronavirus. All right, so some things that we have to keep in mind when we're using antibiotics to use them safely. Certain drugs have certain side effects. Again, hopefully they're minimal. People are different. Some people have allergies to certain antibiotics. That's not, again, because the drug is targeting something in our body. It's because our immune system is reacting inappropriately to the drug. Okay, so like rashes are a common allergy. We'll talk a little bit more about that. Toxicity issues, kidney toxicity is probably the most common toxicity issue, but again, different drugs will have different problems associated with it. So that's something that we'll want to keep in mind. And this disruption of normal microbiota. So this is something that we have a long way to go in understanding, but we're starting to kind of key into this. This is the main reason that we want to use antibiotics as rarely as possible. Okay, we want to keep them for when we absolutely need them and not use them when they're not necessary, because they do disrupt our good bacteria. In our mouth, for example, you have probably at least 200 different species of organism in your mouth right now. A little bit of this, a little bit of that. You probably got a little bit of fungus in there. If you went on an antibiotic that killed some of your variety of microbes and enabled the fungi to overgrow a little bit, then that can give you an infection known as thrush. Okay, one of my colleagues in microbiology, whenever she goes on an antibiotic, she almost always gets thrush. Maybe she has a little bit more fungi naturally in her mouth than what a lot of people do. It's not a certainly very serious disease. We need to go on some antifungals and you get rid of it. Something like Clostridium difficile, C. diff. Okay, that's when people get C. diff. We all have a little Clostridium, most of us, in our large intestine normally, but our variety of microbes keeps it in check. If we go on a strong antibiotic for a few weeks, takes out some of those good microbes, the Clostridium can overgrow, and then we have this really nasty, sometimes kind of chronic bacteria, hard for people to get rid of. It's not just old people. I've talked to college students that have had C. diff. Usually they go on a different antibiotic and are able to get rid of it. People that are older, it can be especially serious. So, okay, so that's something that we have to keep in mind. Ideally, you're going to take, if you have to take an antibiotic, you want to take an antibiotic that will only kill that organism that's causing your infection. Unfortunately, a lot of times we don't know, or we don't take the time to figure that out. If you've got pneumonia, your doctor's not going to say, oh, well, let me see, I'm going to take a week to figure out what you have before we treat it. They're going to give you something that's going to just knock out, whatever bacteria it might be. Okay, so spectrum of activity, is it narrow or broad? A narrow spectrum of activity, a drug is only going to kill maybe a certain kind of organism. Okay, maybe only mycobacterium. That's really narrow. Broad spectrum is when it's going to kill like gram positives, gram negatives, a wide variety of organisms. And again, broad is not necessarily better, because then you're knocking out your good stuff as well. Are organisms susceptible? So if you're trying a common cause of pneumonia, the kind of pneumonia that's often referred to as walking pneumonia, common cause of that is a bacteria called mycoplasma. And what's special about mycoplasma is that they don't have cell walls. So if you give penicillin to try to get rid of mycoplasma, it's not going to work, because penicillin targets cell walls. So the mycoplasma is not susceptible to penicillin. Okay, and sometimes it's because of resistance issues, some acquired resistance. So we can test that by doing something called a Kirby-Bauer test. Okay, so if you are in lab right now, that should look familiar, because we did that a week or two ago in lab, the Kirby-Bauer test. Okay, so if you're not in lab, it looks something like this, where you make a lawn of bacteria. We use a special kind of media called Mueller-Hinton media. And then you put these discs that are impregnated with a certain known amount of antibiotic on that lawn of bacteria. And then we see how big the zone of inhibition gets. And we can refer to charts as to when we measure that zone of inhibition. Well, is that big enough to be considered susceptible or sensitive to that antibiotic? Okay, so in general, if there's a really big zone, then that organism is probably sensitive to the antibiotic. But again, there's some differences as to, the diffusion rate and what we consider sensitive or not. If you have growth right up to the disc, that organism's not being affected by the antibiotic. It's going to be resistant to it. All right, so if a patient has an infection they're having trouble getting rid of, they might do a Kirby-Bauer test and try to figure, put 10 different drugs on there and figure out which one has the potential to work the best. The concentration of the drug you have to keep that in mind as well. So sorry, I have some overlapping letters here. So the minimum inhibitory concentration test, we abbreviate that and call it the MIC test. And typically, if you were screening maybe a lot of different organisms, different concentrations, you might do that in a 96 well plate and have some kind of a plate read your results there. But it's a little bit easier to know what's going on if you did this in test tubes. So you have, you would inoculate these broth tubes with your organism, and the broth tubes would have a certain concentration of the drug in there. So these are getting more and more concentrated here as we go down the line. You incubate it, and then you look to see what's the minimum concentration that worked to kill all the organisms. So in this case, here's our first tube that's not cloudy. So that would be considered the minimum inhibitory concentration because we had growth on all these tubes. Okay, so you got to dial in that concentration and try to get that up to the appropriate amount in your patient. Okay. In thinking about concentration, of course, if you use too much of a drug, that, too high of a concentration, that can be toxic to your patient, but you have to use enough so that it's toxic to the bacteria. So we can express this by something called the therapeutic index. And there's different ways to express this, but that takes the maximum dose of drug that the patient can tolerate, also called the toxic dose, and compare that to the minimum dose that's going to work against your pathogen. So we call that the therapeutic dose. Okay, so a toxic dose is not necessarily what would kill a patient, but it's the dose at which the side effects become unacceptable. Okay. So for example, if you were taking a drug for cancer that makes your hair fall out, well, that's an acceptable side effect because you're trying to save your life. If you take a drug for strep throat that makes your hair fall out, that's probably not going to be an acceptable side effect for most people. Okay, so that's the toxic dose. Sometimes it's easier to express a concentration in terms of toxic dose, test it in animals. But there's usually some variation in between people, so sometimes you express that in an average as well. But let me give you an example here. So the maximum dose of drug a patient can tolerate, let's say that's 100 micrograms, and the minimum dose that stops pathogen growth, say that's 1 microgram. So your therapeutic index is going to be 100. Okay, that's pretty good. That's pretty high. If you had a therapeutic index where you needed 50 micrograms in order to kill the pathogen, then your therapeutic index is going to be 2. Okay, that's not very safe. Okay, where 100, that's going to be a lot safer. Okay, so take home here is the higher the therapeutic index, the more safe a drug is going to be. So higher, the better. Okay, so I will give you a little question next time. That's something that people tend to easily get confused over. I'll give you a little question to practice that. We'll continue with a couple other things here, and then we will start kind of our parade of drugs next time. So I will see you on Thursday. Have a good day.